again, and thanks for having me here. So again, my name is Yaara Arkin. I'm an MSc student at Professor Ran's Halpering Lab at Tel Aviv University. And today I'll be presenting a tool that we developed in our lab, which allows to efficiently detect genetic interactions in genome-wide association studies, specifically in the context of quantitative traits. And I'm going to start by a small spoiler. We ran our method on real data and found two near significant interacting SNPs. And we also compared our method to state-of-the-art tools and saw that in terms of runtime, there's an order of magnitude improvement. So just to start with a quick background to make sure we're all on the same page. So as most of you probably know, genome-wide association studies are a specific kind of genetic study where you have a specific trait at hand, either a disease or a phenotypic quantity. And you want to find areas in the genome which are associated with that trait, and you want to look throughout the entire genome. And you do that usually using SNP chips, which simultaneously test approximately 1 million SNPs. And you do that for hundreds, maybe thousands of samples. So you have a fair amount of data for each experiment. And there are basically two types of experiment that you would do, depending on the type of the phenotype. If the phenotype is binary, such as disease, then you would generally perform a case control study, where your samples are divided into two groups, and you want to test each SNP separately to look for SNPs that might be associated with that division between cases and controls. In this example, it's this SNP over here, where Gs are much more abundant in the cases compared to the controls. The second type of study, similar to what we just heard, is when the phenotype is quantitative. Here you only have one group of samples, and you want to test each SNP for association with that trait. But because the trait is quantitative, then of course the statistical test you perform is probably different. It will be linear regression or ANOVA. And we want to find those SNPs whose genotype is associated with the phenotypic mean. And this type of studies prove to be very efficient in finding thousands of SNPs associated with a variety of traits. These type of studies are specific for testing each SNP separately. But you might assume that the trait that you're studying is affected by more than one SNP. Like in this example over here, where there are two SNPs, SNP A and SNP B, and only if you have, if you're homozygous to the minor allele on both SNPs, then there is a change in the phenotype. And that's not just a theoretical idea. There are many, many examples for this in model organisms. A lot of examples in yeast. Just recently, a study on C. elegans discovered two mutations. That if those two mutations exist in the same C. elegans, you get a 5-fold increase in lifespan. So these interactions are abundant, and in some cases, there's a very substantial change in the phenotypic level. So there's obviously a motivation for searching those type of interactions. However, in humans, only a few interacting SNPs were found so far. And the most plausible cause is just the vast number of hypotheses being tested. If I said that there are approximately 1 million SNP in one study, you have, if you want to test all possible pairs, you have 500 billion hypotheses to test. And that is both a computational burden and a statistical burden. And there are quite a few strategies to reduce that runtime problem. But most of these strategies are specifically targeted at case control studies. If, however, the trait that you're looking at is quantitative, like BMI, cholesterol levels, gene expression, like in the previous talk that we just heard, then there is really not that many options for tools that you can apply. And all of the tools existing are exhaustive. They test each pair of SNP separately. And we'll get to that later, why is that might be a problem. So how would you model an interaction? There's more than one way to do so. I'm presenting one of the common ways. It's used in Plink, for example. And in this case, you have a phenotype Y, and you have two SNPs that you want to test. And you assume that your phenotype is a linear combination of the first SNP, the second SNP, and also the interaction between those two SNPs, which is represented by the product of the first SNP and the second SNP. So you assume a linear model, and your null hypothesis is that this part, the interaction, is not relevant for representing the data. And the alternative hypothesis is that without this third term, you can't properly represent the data. And you would test that using the generalized likelihood ratio test, where you simply compare the likelihood of the data given the null hypothesis compared to the likelihood of the data when you allow the third term to be different than 0. And that test that I just described has to be performed for each pair of SNPs separately. Now comes our tool, EPIQ, which is epistatic detection for quantitative GWAS. And that is the first tool that doesn't require an exhaustive search for all possible pairs. It applies a filtering stage, which finds a subset of SNP pairs which are suspected to be interacting. And those pairs are passed on to validation stage, where they are tested using linear regression. And that is how we gain that improvement in runtime, by not going through all of the possible pairs. So I want to start just with the intuition of our algorithm. If you just look at the data as is, then it's impossible to just quickly find interacting pairs. If you sort the data according to the phenotypic levels, and here you have that each column is a different sample. So now after sorting, it seems like these two SNPs might be interacting. They seem a bit similar. They have similar values when the phenotypic levels are higher. So of course, sorting is not enough for quickly finding interacting SNP. If you have a million SNPs, you would still have to test each pair separately. But this part just gives us an intuition about the similarity that I mentioned between interacting SNPs. What if we could take this problem from this statistical area and shift it to the geometric area, where instead of looking for interacting pairs, we want to look for proximal vectors. So we'd like to find some sort of transformation that transforms each SNP into a vector in such a manner that interacting SNPs are proximal. And once we do that, then we have a variety of tools from the geometric area to finding proximal vectors. So that is actually the core of our algorithm, that shift from the statistical field to the geometrical field. And how do we do that? We do that by simplifying things. First simplification is of the model. Like I mentioned, the full model has 3 terms. But we can ignore these 2 terms, because these 2 terms represent the main effect of each SNP separately. But testing each SNP separately is just doing regular GWAS, and that can be fairly quick. So we can assume that there is no main effect for each SNP separately, so we just want to look at the interaction. So now our model is much simpler. We only want to test whether the existence of both SNPs has an effect over the phenotype or not. The second simplification is simplifying the test statistic. That generalized likelihood ratio test that I mentioned before, under certain assumptions, it can be very well approximated by this new test statistic, tau squared, that we defined. And you can see here that there is a very good correlation between those 2 statistical tests, and that this correlation remain stable even when you reduce the sample size. So now we have a much more simplified problem, and that allows us to make that shift to the geometrical field. So now for each SNP, we convert it into two vectors, V and U, and it's not very important exactly how they are defined for the purpose of this talk, but just notice that we take the information both from the genotype and from the phenotype to create those new vectors, V and U. And these vectors are defined such that for each pair of SNPs, X and X prime, the dot product of the corresponding V and U prime is exactly that tau that we defined earlier. So we are now in the geometric field, and now we wanted to apply a method for finding not proximal vectors, but vectors that have a large inner product, because that inner product is exactly that value of the new test statistic we just defined. So how would we do that? We apply a method of random projections. We sample a random vector R, and we use that vector R to calculate new two scores for each SNP. And what we have now is that for two pairs of SNPs, X and X prime, the expected value of A times B prime is, again, that tau that we want. So what we have, in fact, is a new distribution which is centered around the test statistic. So if there is no interaction, the test statistic is 0, like in the gray line here. So this distribution is centered around 0. However, if there is an interaction, then this entire distribution is shifted either to the right or to the left. And we have now a better chance of passing a certain threshold T that we set. So what we have now is that interacting pairs have a higher probability of passing a threshold. So we now have that filter that we wanted. We can now apply that random projection many times, so that with a high enough probability, our interacting pairs would pass that threshold and would be discovered during the validation stage. So just a quick recap of the algorithm. We sample a random vector R. We calculate A and B scores for all possible SNPs, and we keep them in a sorted vector so that we can very easily discover those top scoring pairs that pass that threshold T. Those top scoring pairs are tested using linear regression. This is the validation stage. And we repeat that enough time to assure a certain level of power, which is given as a parameter for the algorithm. Now we get to the results. In terms of runtime, we compared our algorithm with 4 state-of-the-art tools. EPIQ is here on the last line. You can see that it took only 3.2 hours to run. And for the same amount of data, other algorithms take an order of magnitude longer to run. And also want to note that our algorithm runs on a regular hardware, and it's still faster than these two algorithms which run on a dedicated hardware of GPUs. So maybe you could imagine that if you run our algorithm on GPU, it might be even faster. That's in terms of runtime. In terms of real data, we applied our algorithm to data from the Lurie cardiovascular health study. The phenotype was lipid concentration in cells, and our algorithm ran for 1.5 hours on 10 processors and found 2 near significant SNPs on chromosome 6 and on chromosome 5. So to sum up, what I just introduced here is the first non-exhaustive quantitative tool for finding genetic interactions in GWAS. And that non-exhaustive part is important because if you want to think about possible extension, it's possible to imagine a way of extending our algorithm to interaction between 3 SNPs. However, if you want to extend an exhaustive tool for trios, it's impossible in current day hardware. Another possible extension that I think might be very interesting is maybe applying this method to gene-environment interactions. And of course, I want to thank my colleagues, first of all, my advisor, Yaara Arkin from Tel Aviv University, which worked on the algorithm with me, and Eliyahu Rahmani, which worked on the data, helped process the data, and the lyric study team, which supplied the data. And of course, you. Yaara. Yaara.
Okay. And then let's have questions from both speakers and have a little discussion.
Interesting talk. So I have a few questions. From your algorithm, it seems that maybe I missed something. You try to find pairs of SNPs that actually are the vectors that are highly correlated, right? Which means they may have, if two SNPs have a strong LD, right? Would you find them as a candidate pair?
Yes, that is a very good question. I didn't get into it because of the time, but if you see in our paper, then you can set a certain parameter of what is the minimal distance between a pair of SNPs. So if you have SNPs which are too proximal, our algorithm will dismiss them, and that is our method of overcoming that problem of linkage disequilibrium.
Well, even if two SNPs not proximal physically to each other, but they may still sort of like have LD, and and then which means that if one of them have strong main effect, right? Because the other one is almost similar. I mean, it's very similar to this. Then you would detect them as a pair. But I have the question, like, because when you filter your candidate pair in that way, you may miss a pair where they don't have a strong LD, but together actually their interaction is strong. Their joint effect is very strong to your phenotype. Would you be able to find those?
Well, currently this algorithm just performs the linear regression, a simple linear regression. But of course, it's currently a beta version, so there's always possibility of refining it.
Okay, so another question I have is, maybe you didn't have time to talk, is how do you control false discovery rate?
Currently, we just apply a Bonferroni correction.
But that's conservative. Strict. It's very conservative,
yeah,
right? Especially if a large number of tests. Yeah.
Currently, that's what we perform. It's a work in progress.
And I would also recommend you to look at a few, maybe paper published 5 or 6 years ago at the RICOM and the ISMB, and their works on speeding up those interaction detecting.
For quantitative data.
So there's a series of algorithms I can discuss with you offline.
Okay, I'll be.
Their works on not doing exhaustive search, but still guarantee finding the answer with a controlled error rate. Yeah.
Thank you.
Yeah, I think your method is assuming that you have an interaction between the minor alleles, because I think only in that case the vectors will point in the same direction. What if you have an interaction between a minor allele on the first locus and a major allele on the other? I think in that case the vectors would actually point in opposite directions.
Yeah.
Could your method account for that?
Well, of course, that's also dependent on how you encode the genes, because in our algorithm, part of the simplification is a binary encoding of the SNPs. So you could, for example, try to encode both a minor allele encoding and maybe the opposite encoding, and try to find the possible combinations. So in this case, you would increase the number of tests that you perform, but you will cover all the possible options.
I think in that case you would have to do an exhaustive search again, because you would have to flip labels of each pair of markers.
Oh, you can.
Maybe this is too detailed, but I don't think it's that easy. Maybe another question.
There is something similar in the rapid algorithm. which which works on case control studies, where they encode the SNPs in 2 different ways, and then you have also the minor allele effect in the.
Okay. Another question was, maybe you were.
Can I just jump in a minute?
Yeah.
Benefit of the audience, because you have 2 questions that kind of relate to how the methods handle acquisition. Right? You're asking about linkage to disequilibrium, and you're asking about positional coding. So I want to make that point a little more clearly, and now come back to both speakers and ask about that position and how you deal with that explicitly. Because this is potentially a source of bias, right?
I'm sorry, I'm not sure I understand the question.
I think I'm being clear, but maybe I'm making it worse.
You're right.
Thank you. Okay, so let's talk about these 2 methods from the standpoint of how they handle the position of the SNP in the physical map or in the sequence, okay? And you're saying we don't have to worry about it because we're not encoding the map position, right? Did I understand you that correctly?
Encoding the position on the genome?
The position of the SNP in the sequence, right?
Yeah, we
Right, you're
don't.
not encoding that.
No.
And then the worry comes, but what about linkage disequilibrium? Because linkage disequilibrium biases the transfer of adjacent markers in a particular way. So you guys, I think if I understand correctly, handle the linkage disequilibrium, right? And you do not? Do I understand this correctly?
No, currently we do not. We, since most of the SNPs in linkage disequilibrium are proximal, then you could, for example, test that's a linear number of pairs, and you can test them separately beforehand or afterhand.
But my question for you is, if you're not encoding position and you see a higher rate of association between 2 SNPs, then either that's due to disequilibrium, linkage disequilibrium, or it's due to an actual bona fide association. So how do you distinguish those 2 cases?
The problem of to avoid the problem of too many false positives, because almost all proximal SNPs might seem associated, then these proximal SNPs are disregarded. We do address this. But if there is linkage disequilibrium in distal SNPs, then this would have to be tested in a different way.
So you go back and you check for adjacency of SNPs.
Yeah, before the validation, before we start validating, if you say if they're proximal, then we disregard them.
And now could you restate how you handle linkage disequilibrium to the benefit of the audience? And maybe you should use as loud enough as I am.
So in our work, actually, it's my student who did the presentation. So the way we could handle the linkage equilibrium is that that information can be encoded as a graphs of SNPs, where in the graph each vertex is a SNP, right? And the edge can encode the weight on the edge would be the LD information, and that can be treated as a prior information as an input, because even though in his talk he just said 1S, 1G, but there's no limit how many such graph we can incorporate as prior knowledge, and there's no need that to be very precise and no need to be complete. Yeah.
So maybe you guys ought to hybridize. We've got time for like 2 more questions. Go right ahead. That was a wonderful triggering question.
Thanks. Just a really quick question. I think it's easy, and maybe you were saying it and I just missed it. So if you have a marker where you have a very low minor allele frequency, then in this vector you will have few ones. If you have a large minor allele frequency, you will have many ones. So the vector norm will depend on the minor allele frequency, and I would assume you would have to account for that by essentially just looking at the direction but not the length of the vectors. Is your method doing that?
I actually skipped this part because it would take longer than the 20 minutes that I had, but we perform binning of the SNPs according to their minor allele frequency.